Welcome to another episode of Progress, Potential, and Possibilities, discussions with fascinating people designing a better tomorrow for all of us. I'm your host, Ira Pastor.
Welcome, everybody, again to another episode of our show, bringing you another really awesome guest today involved in creating a better tomorrow for many people out there. Today, we have the honor of being joined by Craig Thompson, who is the Chief Executive Officer of Cerevance, which is a private clinical stage drug discovery and development company. They're focused on developing novel therapeutics for CNS diseases using their company's proprietary NET-seq technology, or nuclear enriched transcript sort sequencing technology, a really interesting machine learning platform that is involved in measuring really precise levels of thousands of genes expressed in various cells in different regions of the brain, ultimately mapping those genes to different disease and healthy brain states to be used in drug discovery and development, but also in some really interesting areas that we haven't really gotten into as much on past episodes, areas like patient monitoring. Most recently, Ms. Thompson was the president and chief executive officer of both Neurana Pharmaceuticals, which is a biotech company focused on neuromuscular conditions, also CEO of Anthera Pharmaceuticals, which is involved in developing and commercializing a range of products in the area of cystic fibrosis, inflammation, autoimmune diseases. And prior to that, he had some time as chief operating officer at Tetraphase Pharmaceuticals and chief commercial officer for Trius. And he was involved in their acquisition by Cubist, also in some major partnerships with Bayer. And prior to that, he served in various global and U.S. roles at Pfizer, including as Therapeutic group leader of Allergy, Respiratory, Pulmonary, Vascular disease, and inflammation, as well as vice president of marketing for Pfizer's specialty care business group. And before Pfizer, was involved in various positions of increasing responsibility at both Merck as well as with their partnership with Schering Plough. He's got a bachelor's degree in commerce from McMaster's University, his MBA at Notre Dame. A lot of interesting themes to get into today on the drug discovery and development and AI front. We're honored to have him. Craig Thompson, thank you so much for taking the time to come on the show.
Thanks, Ira. It's a pleasure to be here. I'm looking forward to sharing what we're working on at Cerevance.
I'm very much looking forward to it. It's an exciting set of technologies, and obviously you've gone from a platform company into what looks like it's going to be a fully integrated shop shortly. But before we get into all that, would love, as we typically do, to hand you the floor for a few minutes just to further introduce you. I noticed in your time at Big Pharma, you were involved in commercializing some of the major blockbusters at some of these companies. Tell us a little bit about your background story, what got you into pharma, and a little bit of what made you decide to get into smaller biotech to create a pharma ultimately.
No, it's a great question, and I started my career way back when. I won't go into the number of years, but at Merck, which was an amazing experience. So I spent almost 12 years at Merck and started off in the vaccine group, really kind of understanding, how to build up a vaccine division based on Atlanta, and did a lot of work with CDC, and at that point was for Recombavax and all the OSHA requirements for. You should probably give me a little bit of flashback to when that happened. So it was a great experience, and always took the I think the unusual path. So like a lot of people in pharmaceuticals, did a brief role as a sales rep in New Orleans and Shreveport, Louisiana, and then went straight into the global group working on anti-infectives. So always have found kind of interesting opportunities. Always worked on products that I was passionate about, and felt like they made a difference. Nice thing with Merck was the entire portfolio was based that way, so you couldn't really go wrong. But it really gave me a really nice opportunity to kind of understand different products, different disease areas. So I spent a lot of my time at Merck, both on the global side as well as the US side, mostly on the anti-infective sides, working on a lot of products that were still in development.
Mhm.
But also had the opportunity kind of later in my career at Merck to work on the Merck Schering Plough partnership for the launches of Vytorin and Zetia, the
Yep.
cholesterol-lowering medications for Europe, the way it was set up, which gave me kind of an experience into a different company culture after being at Merck for 10 years to see how Schering Plough was at that point a separate company. And then kind of out of the blue one day, I've got a call from a recruiter who had a opening at Pfizer, and thought, why not? Always had the dream of living in New York City. I grew up in the suburbs of Toronto, so living in a big city was always kind of what I wanted to do. So had the opportunity to move to New York City and work for Pfizer. First, actually, in a partnership. Working on a respiratory drug, and then continually kind of moved ahead. And I think I always brought my style of kind of transparent, open communications of if you see a problem, call it a problem, figure out how to correct the problem. But unless you actually call it what it is, it's really hard to fix. So I always kind of brought that kind of sense of kind of honesty and transparency to the way I've moved my path forward. And had the opportunity at Pfizer working on several drugs. So one of them was Torcetrapib, for
Yep.
example, which was supposed to revolutionize Pfizer with the HDL. And that one frightful Saturday morning, got the call that we did see a mortality imbalance, but worked tirelessly over the weekend, and that was a product that we actually won the Investor Relations Award for kind of best investor relations crisis. Not something I'm particularly proud about, but I think it goes to the core of again, we saw a problem, we quickly acted. Stopped the clinical trial within 12 hours. We had all the patients off drug. So just very quick, very decisive, make decisions, move forward. And then later on, as you mentioned, had the opportunity to head up allergy, respiratory, pulmonary, vascular disease, and inflammation. A little bit of an odd conglomeration of different groups. I started off with allergy, respiratory. They needed someone to help head up pulmonary vascular disease, so I picked that up, and then I picked up inflammation as well. So did that for a couple of years, and that was fantastic because I was able to not only see what we were doing from a commercial perspective and work with our great partner Boehringer Ingelheim on Spiriva, but also be in with some of the bench scientists over in Sandwich, UK, when Pfizer still had that site. So great opportunity. And then from there, moved into a straight commercial role as VP of marketing for specialty care in North America. And it was another kind of great opportunity looking at the specialty care products, a lot of more hospital focused. We did the Wyeth acquisition. At that point, we were moving, oddly enough, specialty care from New York City out to Pennsylvania. And love Pennsylvania, but had already lived in Pennsylvania and didn't want to kind of repeat that part of my life again. So I spent a year going back and forth with from New York City to Pennsylvania and into the Wyeth site. And at that point, one of the people I actually worked with at Merck was at a company called Trius, and called up and said, "Hey, we're looking for someone that looks like this. Do you know anyone?" The timing worked out really well, and from there I went to a small biotech. So I'm from, managing a few $1 billion to, a very large group, to being, I think I was the 35th or 36th person on the team. And I remember my first introduction to the company was, for the people listening, probably know JP Morgan. Was the night before JP Morgan. Met our CEO, Jeff Stein, and he passed me a cell phone and a computer and said, "You're starting tomorrow." And that was my introduction into biotech, was day one at JP Morgan. So it was a great experience. Had a great team at Trius as well. Jeff's a dear friend, a great mentor. Really helped me kind of learn that transition from big pharma to biotech, and no regrets since then.
Excellent. No, really outstanding. I enjoy listening to a story like that and to see sort of the degree of skills and sort of new experiences, that you gathered up along the way, which obviously makes you perfect for what you're doing now because it sort of all comes together in sort of the full spectrum of. And I was I was reading a bit about you before the show, and you're talking a little bit about the stuff you're doing now, and that a lot of these AI or machine learning companies, position themselves sort of solely as sort of a platform, but you've gone far beyond that in the sense that, you're not just discovering sort of a new chemicals, but you're developing them, you're going into clinical development. And the core of what this company is all about, as I mentioned in the bio, is this net sequence, technology, nuclear enriched transcript sort sequencing. Would love to take you, for you to take us into a little bit about sort of the background to the technology. Obviously, these models require lots of input. So in your case, you have samples, thousands of them, gene expression profiles. Clearly, the drug development side. Walk us through sort of the beginning or sort of the inception of this tool, and a little bit about how the company sort of in the early days sort of evolved from just being this tool company to, hey, we're getting some good hits here, and then we'll get into some of the candidates a little later on that you have in the pipeline.
Sure. Yeah, so it's a really, really interesting tool that came out of Nat Heinz's lab at the Rockefeller. Um, so Nat had previously developed a system called Backtrap, which used transgenic mice. And I won't go through that process because it's kind of an upgrade to that. But I think what really makes this unique. So everyone's very familiar with single cell sorting, and this is not that. We use single cell sorting. Single cell sorting is great. So don't take what I'm going to say as a kind of knock on single cell sorting. We just think that this is gives us a little bit more depth into exactly what is going on. So we use postmortem human brain tissue. We work with 26 brain bank partners, mostly in Europe, US, the UK, to get samples of disease areas that are of interest. So obviously we have a nice collection of things like ALS, FTD, Parkinson's, Alzheimer's. But along with that, we also get about half of our samples are from patients that didn't die of brain disease. The reason why that's important is as we process it, we want to make sure that we can take out one of the biggest confounding factors, which is age. So we have patients from 8 to 104 years old in our kind of control sample, so that we can really look and try and case match and look at what is exactly going on. I think what makes NetSeq very different than a lot of other ones is that we don't actually sort the cells. We get the cells. We then extract from that the nuclei, and then we use different riboprobes to tag those, and then use FACS sorting. From that FACS sorting, we then get pure populations of nuclei, which are tagged, and then we sequence those. So from a cell, we'll pick up about 12,000 genes, which is a lot. And we can sort about 60 different cell types. So you start doing the math in your head, you're like, wow, you guys are picking up lots of data. We've actually sequenced so far north of 60 trillion base pairs. So as I always like to say, we don't use AI machine learning because it's kind of the cool buzzword. We use it because we have a lot of data. And we need to be able to interrogate that data appropriately. But back to NetSeq just for a second. So we get the data, we sequence it. That then gives us an idea of what's happening in a single subject at a particular time. But we have a collection of patients that have, or subjects that have the disease at various stages of the disease. So Alzheimer's, some will have it end stage, some will get in early stage Alzheimer's, some at mid stage. We then layer on the machine learning so that we can actually use what we call pseudotime analysis and start mapping out by cell type what gene is either upregulated or downregulated. So that then starts to give us very specific information as to which group do we want, what cell and what gene do we actually want to go after. So we spend a lot of time understanding what the target is. Once we've understood what the target is, we then go into the next page, which is the chemistry of, how do we develop a compound to actually either antagonize this or agonize this particular gene. The other nice thing is that because we can get so specific, we can look at, so if you look at one of our later stage compounds, it's a GPR6 inverse agonist. GPR6 is only expressed in the medium spiny neuron of the indirect pathway. The reason that's important is we're able to push the dose because we're not going to have other on-target side effects because it's not expressed anywhere else in any significant way. So it starts to give you a little bit of insight. What can you do from a dose selection perspective? It gives you a little bit more flexibility because you're not going to have other on-target side effects because you're very selective. So that's great. And we've also recently started putting overlaying GWAS data as well. So we're starting to get genetic links. And we're using ATAC-seq as well, looking at the open and closed chromatin. So we can start seeing exactly what are the regulatory regions. And a good example of that is one of our earlier targets that we haven't disclosed what it is, but the literature actually describes that particular SNP associated with the closest proximal gene. And we have actually shown that it's actually not associated with that gene as the regulatory one. It's associated with a different gene. So it starts to give a more understanding of exactly what's going on from a biology perspective. So with all that information, then go into the chemistry side of it and try and develop compounds. We tend to do more small molecule. Part of the reason is all these molecules need to be brain permeable. So they tend to be high solubility, high permeability, low solubility, got that backwards, sorry, to get across the blood-brain barrier so that they can get to the site of action.
So just to repeat in terms of this lead candidate, which is known as CVN 424, novel, first in class, it's potent, it penetrates the brain, it's not working on dopaminergic system, which is sort of been the mainstay of pharmacotherapeutics in the area for a century now. GPR6 inverse agonist, really cool. And you've gone as far as positive phase 2 results. Talk a little bit about, take us into the clinical work, because obviously, it's all come together, right? So the biology, the machine learning, you picked a great candidate, you made the transition into clinical development. Talk a little bit about what you saw so far in the phase 2 results, and a little bit, just if you can say a little more about when you have something like this that is very new and novel. It's such a novel target compared to where we've been with the Parkinson's. A little bit about sort of what the triage process for you internally was, that hey, this is this is where we're going to stake our claim and move with this into the clinic.
Yeah, I think a good way. So we were looking at Parkinson's and Parkinson's agents, and we were pretty excited about going through the non-dopamine pathway. So up till now, with the exception of Neuroscience, everything's gone through the dopamine pathway.
Right.
Either CMT inhibitors, MAO-B inhibitors, basically increasing the amount of dopamine or stopping the degradation. You have dopamine agonists, basically tripping the dopamine receptors. You have levodopa, giving more exogenous levodopa. So we were like, is there a way to actually treat Parkinson's by not going through this particular pathway? And if you think about Parkinson's, it's controlled. If you think about movement, it's controlled by the indirect and the direct pathway. And the best way to think about this is if you think about a hydraulic arm that's moving, you want to have that kind of constant pressure of kind of pro movement and then braking movement. What happens in Parkinson's is that gets out of joint with lack of dopamine being produced. So you get the direct pathway, which controls the pro movement, if you will, and the indirect pathway is braking. So in Parkinson's, what happens is that indirect pathway becomes hyperactive. So you get kind of this like severe slamming of the brake on, and the direct pathway, which is promoting movement just doesn't have enough gas. So if you're at a stoplight, for example, and you're sitting there and you don't have your foot on the accelerator and you have your foot on the brake, nothing happens. That's kind of what happens in Parkinson's, if you will. So we were thinking was there a way to actually go after the indirect pathway, not through the dopamine pathway? And our biologists were looking through and found this unique GPR6, which controls the signaling of the indirect pathway. So typically what happens in Parkinson's is the body receives the dopamine, dopamine binds to D2, D2 then downregulates cyclic AMP, which is the signaling mechanism. What we're actually doing is we're actually stopping that production of the cyclic AMP.
Okay.
So, the way I always like to look at it is, very novel, but, if you have a hose and a faucet, the dopamine tube basically comes through and squeezes that hose. GPR6 is what's actually producing or helping produce that cyclic AMP, and we're turning that faucet off. So you have less need for the dopamine actually to come in the side path. So when we were developing this, one of the things we wanted to make sure was that the efficacy was going to be at a place that would be significant for patients. So if you look at a lot of the recent launches that have happened, they haven't done particularly well. So most companies have looked at their products for adjunctive therapy, so on top of standard of care. Most of them have about an hour improvement of off time. And in Parkinson's, the lingo is off time when you can't move, on time when you can move, and then on time with troublesome dyskinesia. Dyskinesias being those kind of rapid flings. Think Michael J. Fox. Yep. Great with a kind of rapid flings or sleep. And what you really want to do is move those patients from off when they can't move to on when they can move. Right. But not put them on dyskinesia where they have those rapid flings. So you're trying to bring back normalcy to their life. Yeah. Most of the products that are available right now, including recent launches, had about an hour improvement in off time, which doesn't sound like a lot, but they are on background medications, sure. And it is pretty significant. Where you start to see even more improvement is when you go up to some of the infusions, like AbbVie has Duopa. It has about a 2-hour improvement. That unfortunately requires a pump that you wear with you, includes surgical infusion. Also working on another novel product, ABV 951, which is a subcutaneous delivery as well. That also requires a pump. But patients are willing to go up to using a pump, if you will, to get from 1 hour to 2 hours. And then the next step in that cascade is deep brain stimulation. Exactly what it sounds like. You put probes in the brain, and then you go, which are surgically implanted, and then you go back to the surgeon's office a couple of weeks later, and you have a battery implanted into your chest so you can regulate the stimulation of those probes. And that gives you about 4 hours. Obviously, no placebo-controlled studies for that. So we wanted to make sure that we had a product that was going to be well above an hour improvement in off time, because everyone else has had an hour. Market doesn't seem to get particularly excited about it. Patients don't get particularly excited. We wanted to get closer to that 2-hour mark. So what we did on our phase 2 study was we ran a relatively short study. So most of the regulatory approval ones for phase 3 are 12 to 13 weeks long. We ran a 27-day study. Reason being is we wanted to see if this was going to have kind of the horsepower over a month to make a significant impact. It wasn't going to do that, no sense in moving it forward. Right. One of the things we found was from baseline, we had about a 1.5-hour improvement in off time. So above where we're seeing the other orals, and then almost up to the effect that we're seeing with the infusions. So in discussions with some KOLs, one of the learnings for us was the FDA analyzed the data slightly differently. So they suggested that we analyze the data in a slightly different way and use the standards that the FDA was going to subject us to for phase 3, which was they want to make sure that all the diaries are filled out correctly. And the good news was all of ours were, except for one. But they also normalize all the patients to a 16-hour waking day. The reason they do that is Parkinson's patients tend to sleep. And then as you give more medications, they tend to get sleepier. So they want to make sure that you're using a standard benchmark, which is a 16-hour waking day. When we do that analysis, we see about a 1.78-hour improvement in off time, placebo adjusted. Again, now it's starting to get really close to what you're seeing with infusions. So that was pretty exciting. The other point we also saw that was exciting, and we saw this in our preclinical models, was impact on non-motor symptoms.
Yep.
So if you think of apathy, depression, sleep, we've run a lot of preclinical models, and we can show that mice, we get that drive back, but we weren't sure if that would happen in humans. It should, but we did see an improvement in the UPDRS Part 2, which is the Unified Parkinson's Disease Rating Scale patient report outcome. So are you feeling better? Are you feeling more motivated? And ask a series of questions like that. And we saw almost a 3-point change in 27 days. Generally, you're looking for about a 3-point change over 12 weeks. So you're seeing a pretty important impact. The other one that was pretty dramatic was the Epworth Sleep Scale. So patients were modestly sleepy, so they had a baseline of 10 to 12. When they were taking the high dose, we saw about a 2-point improvement, so about a 20% improvement. So you didn't see what you normally see with Parkinson's, is you gave them additional medications, they actually were more alert. So completely different than what you normally see. So patients are getting, at least from the phase 2 study, better improvements in off time. They're having impacts on their non-motor symptoms, so they're feeling better, they're saying they're feeling better, they're more alert. The last question is, was the product well tolerated? And the simple answer is, in the study, it was well tolerated. The only AEs that we had in greater than one patient was nausea, vomiting, and headache in 2 patients, or 4%. So we didn't see a lot of the typical adverse events that you see with products like the dopamine agonist, CMT inhibitors. You don't see any of those dopamine-related side effects like hallucinations, orthostatic hypotension, dyskinesia. I always kind of joke, probably inappropriately, I'm like, those would be great Friday night events to have, but not necessarily your kind of everyday life. So it was really nice that we weren't seeing any of those adverse events, and we didn't anticipate seeing them, but you don't know until you put those in clinical trial. So that was the first study we did. And then right now we're conducting a study in monotherapy. So this is really novel. So the last product that was approved for monotherapy was Sertraline in 2006, so almost 20 years ago. Tavapadone, the Cerevel product, is also looking at monotherapy as well. So it's a dopamine agonist targeting D1 and D5. So we're kind of following the same trajectory as that product. But the idea there is to try and treat patients and delay the initiation of levodopa. Because once you start levodopa, you go down a very predictable path of, you start on levodopa, you go from off to on, you then increase the dose of your levodopa, you then take it more frequently. Levodopa has a 90-minute half-life, and you start to get in the sinusoidal wave of off to on troublesome dyskinesia, and it just gets bigger and bigger and bigger. They're very predictable what's going to happen. So if we can show that 424 can delay the initiation of levodopa, that could have a meaningful impact on patients' lives, not necessarily at the beginning, but when they're later in their disease, really delaying that initiation of levodopa and delaying those fluctuations from on to off. So that study is currently enrolling. Pretty simple study, actually. We have 60 patients that we're going to enroll, 30 in placebo, 30 in the high dose. They can't be on any background Parkinson's medications. They're treated for 12 weeks. And then at the end, we measure the UPDRS, or Unified Parkinson's Disease Rating Scale, Part 2 plus Part 3. Part 2 is the patient-reported outcome. Part 3 is the physician assessment of the patient. It's also, in that study, we've also brought forward some, what I think are really interesting digital technologies, and some of them use AI, for example. One is a group out of the Bay Area called Modality. Modality, much like we're having this discussion today on Zoom, you speak into an avatar on either iPad or a tablet, and the avatar, as you're speaking to it, is asking you certain questions. But at the same time, it's mapping out your facial structure. So it's looking for any changes in cadence. the word choice you use, the volume you're using, your facial expression. So generally, as Parkinson's progresses, patients tend to be what's known as mask-based, so they get very
Right.
blank. They tend to speak softer. They tend to use simpler words. So this will be measuring all that, and we'll have it at the beginning of the study and the end of the study. Obviously, we'd like to see with 424 no deterioration, and placebo we'd like to, well, wouldn't like to see, but we expect to see some deterioration of it. So that'll give us a really interesting look into the patient. Again, that uses a lot of AI algorithms to help analyze the data because it's picking up a lot of data. On top of that, we're also picking up verbatims from the patients of, how are they feeling? What are the words they're using? What are the word choices of how the medication's impacting them? We're also using Cogstate, which a lot of companies use for CNS.
Right.
And then the third is we should have an impact based on the Epworth Sleepiness Scale on sleep and sleep architecture. So we partnered with a company called Beacon Biosignals, and they use an EEG monitor to look at EEG waves so we can start looking at that sleep and sleep architecture. I think what makes this particular aspect or this particular application unique is, and they probably not like my description, but it basically looks like you put a bandage on your forehead.
Mm-hmm.
It's a little bit bigger than that, but that's what the patient needs to do every night. They just put this little strip on their forehead and they go to sleep. So there's no wires and monitors hanging out, but a little strip you put on your forehead, and then we'll pick up all the EEG data as well, so we can start to see what impact are we having on sleep, and hopefully having a positive impact on sleep.
Mm-hmm. Mm-hmm.
So that's pretty exciting as well. And then right behind that, we're just planning our phase 3 study, which we anticipate to be very similar to the phase 2 adjunctive study that we ran.
It's extremely intriguing that that you're using this core AI tool to not just again in drug discovery and development, but you're also creating these companion diagnostic tools to go along with everything in the clinical trials, and as you're talking measuring gait and masking and all these other motor and non-motor
Yep.
components of the disease. It's extremely elegant, and really again speaks to the way that Cerevance has thought through sort of kind of a holistic approach of this platform to sort of all parts of the chains. It's extremely impressive.
I think part of it comes from the idea of, and I know a lot of people say this, but trying to be as patient-centric as you can be.
Right. Mm-hmm.
So obviously there are the regulatory endpoints of, for an adjunctive therapy, we need to improve off time.
Right.
Very cut and dry what we need to do. That's great, but there's so many other subjective endpoints. So we've looked at a lot of different technologies to try and implement, and at one point we were looking at other ones which were probably a little too invasive, but we really want to understand how is this treatment going to in fact impact patients and patients' lives. Can we delay the initiation of levodopa? Can we delay their disease to move it forward?
Mm-hmm. Mm-hmm.
And really try and look at it from the perspective of the patient. One of the things I always ask my team to do, and I say generally it gets done, is when we're putting together protocols, is run yourself through that protocol.
Yep.
What's it like when you go to the physician's office? And I've been at companies. One of the companies I went to that you mentioned in your intro. When I joined, they were just about to kick off a phase 3 study. And I looked at it, I was like, there's no way a patient's going to do this.
Yeah.
You put 18 measurements in there, which is great, but it's going to be 3 hours before this patient's actually filling this out and they're in pain. It's not going to happen. Like the data you're going to get is bad. So always put yourself in the mindset of what is that patient experiencing in that moment, so that you can try and get the most relevant data and the most appropriate data that's going to help you differentiate the product, so that patients and physicians and payers find it useful.
Mm-hmm. Mm-hmm.
And there's always trade-offs,
Cool.
but it's it's usually a trade-off that I don't think a lot of people think about. Of if I have a patient in there who's 2 hours into the visit or 3 hours into the visit, what is that quality of the input that I'm getting? When I went through this one example, I was 2 hours into. I was just checking boxes. I was like, how do I get this done?
Right.
That's what patients are going to be like as well,
Right.
and they're not as committed as you if you're practicing doing that.
Craig, I know you mentioned, you have several other assets in the pipeline. Obviously, the Parkinson's one is the most advanced. I was wondering if you could just say a few words about another one that kind of stood out to me is really impressive. It was CVN 293, and I think you're developing this for both Alzheimer's and ALS. But what intrigued me here most is that your target, or again, this goes back to sort of the gene expression profiles of, within the NetSeq technologies. But you're looking at the inflammasome, sort of, all things happening neuroinflammation wise that lead to neurodegeneration, and obviously. Our industry has been spending a lot of time upstream with beta amyloid and all that stuff. But say a few words about this one, because I think it's you're looking at a totally different area, but one that I think most people would agree nowadays, we got to stop this destructive, inflammation, cell death, and ultimately degeneration. Can you say a few words about CVN 293?
Yeah, it's a really interesting target. So our team started working on this target about 4.5, 5 years ago. And I think in the literature at that point, there was probably a dozen publications even describing the target, which is KCNK13, a potassium, 2,4 potassium channel.
Mm-hmm.
So I think within the scientific community, there's general agreement that inflammation is bad.
Yeah.
And generally, when we look across companies that are working on the inflammasome, and particularly the NLRP3 inflammasome, which seems to be the one most companies are focused on, they're really approaching it from the periphery, and really trying to kind of wipe out or really significantly knock down the NLRP3 inflammasome, so that it can have some central benefits, and that's usually working through macrophages. Our thought was, it's probably going down the right path, but a little misdirected. And a science, so we'll find out what the reality is, obviously in the future. But our approach was, how do we modulate the NLRP3 inflammasome and still leave the inflammasome open to what it's doing, and making sure that there are parts of the inflammasome that we like to keep, and parts that we obviously want to dampen down. So we wanted to look at it more through the microglia, and how can we focus on the microglia, and then was there a way to dampen this down? Because what you want the microglia to do is, there's surveillance, motility, and chemotaxis and phagocytosis that you still want to happen, but you want to tamp down that neuroinflammation. So what we found was KCNK13 early on in the activation of parts of the NLRP3 inflammasome. So by knocking down that 2,4 potassium channel, we can stop the inflammation part of it, but still leave the other components that you'd like to happen with the microglia. Nice thing is we get very high expression centrally, very low expression in the periphery. So if you're thinking you want to work in centrally, that's exactly what we're doing. We're not touching the periphery. So if you're an elderly patient and you think about the patients that we're potentially treating with this product, with ALS, Alzheimer's, they're probably going to be more elderly, probably a little bit more fragile. So if they get an infection, you still want the inflammasome or the inflammation system to react to the infection, but you want to tamp it down centrally, so that you're not getting that neuroinflammation, getting that cascade of inflammatory effects in disease like Alzheimer's and FTD, ALS. So this product has moved relatively quickly. One of the things that we did see with our AI machine learning was that the target KCNK13 is actually overexpressed in patients with Alzheimer's versus slightly underexpressed in patients without. So we start to see it going up, and we see this both at end, and so with healthy or patients that didn't die of brain disease and those that died with Alzheimer's. We also, when we use our pseudotime analysis, we see a nice upward trajectory as the disease progresses. We also see higher expression of it as well. So the preclinical models gave us really good insight into the product working on the inflammasome. And currently we're in a phase 1 study on a single ascending dose, multiple ascending dose, currently dosing cohort 5, so we've obviously moving up quite nicely in the dosing range, and just started our multiple ascending dose the past couple of weeks, and we should have data from that at the end of Q1. What I think is particularly exciting about this compound, not only what it potentially could do for ALS, FTD, Alzheimer's patients, but in parallel, we're also developing a PET ligand.
Okay.
Which should help us from 2 perspectives. One is it will help us from a target engagement perspective, but hopefully it will also help us from microglial inflammation. So right now, the only kind of compound we have for microglial inflammation is TSPO, which is not particularly effective. So the PET ligand could actually be a very useful tool beyond our program to other programs looking at microglial inflammation. That's a pretty exciting kind of, I don't want to say side project, but additional project that we're working with it. So ideally, once we finish this SAD/MAD study in end of Q1, beginning of Q2, by the end of the year, we could potentially be looking at a study in more than likely ALS, FTD, with a PET ligand, so we can actually start to look at that microglial inflammation and target engagement in patients and show that change over time. So taking a very different approach than everyone else in the industry, which we believe is the right way to go, and provides a lot of benefits that others are looking at without a lot of the risk profile. And the nice thing is with KCNK13 is it's predominantly expressed in the microglia. We don't see high levels of expression anywhere else. So again, it gives us the opportunity to try and push the dose a little bit because our on-target side effects should be limited to microglia.
Impressive. I mean, just the exciting mechanisms and the speed by which you're moving everything forward is quite impressive to get this human data. And clearly, you've brought. I mean, it was last year you closed a Series B at $116 million, and you have a rather diverse range. I mean, you have the corporate venture funds, you have traditional venture funds, you have the venture discovery fund, and the Gates. A lot of exciting investors bringing money to the table. They clearly see the potential of what you're doing here. What else is coming up that we should know about, Craig? I guess you just got back from JP Morgan probably
Right.
a couple weeks ago. What's happening as we get further into 2024 that we should know about as far as you and the company?
Yeah, so a couple of really exciting things. So, as you mentioned, with the Karuna and Cerevel acquisitions, we definitely have a lot more interest in CNS of late, which is great. Us moving forward, just from a timeline perspective, on CVN-293 that we mentioned, we'll have data from that in end of Q1, beginning of Q2 from a SAD/MAD perspective. The monotherapy study I mentioned for CVN-424. We'll have data from that end of Q3 as a monotherapy. So those are kind of 2 very exciting data points for us. At the same time, we also have 2 preclinical assets that are disease modifying for Parkinson's in the preclinical stage right now. So those potentially could enter the clinic the end of 2024, which is pretty exciting.
Right.
And then at the same time, we also have a program that we didn't speak about. It's an orexin-1 antagonist. So it's one that we're actively looking to out-license. Not that we don't like the program, we actually love the program. It's just we're focused a little bit more on neuroinflammation than neuropsychiatry. But it's a really interesting compound. We're about 1,000-fold more selective for orexin-1 than orexin-2. Why is that important? Merck has a product, for example, suvorexant, works on orexin-2, or it's Adora, works on dual orexin antagonist. If you hit orexin-2, you work on the sleep center. If you hit orexin-1, you're working on the reward center. So our product, because it's 1,000-fold more selective for orexin-1 in our phase 1 study, we're able to get really good receptor occupancy and not see any signs of somnolence. So avoiding that orexin-2, so we're just not seeing that sleepiness. But that product has potential to work in, since you're working through the reward center, things like negative cognitive symptoms, schizophrenia, Prader-Willi, binge eating, any of the addictions because you're trying to tamp down that reward center. So
Right.
pretty exciting product from that perspective. So we are having some active discussions on potentially out licensing that product.
I have no doubt you'll find a lot of partners interested in that one too.
It's
No,
pretty, like I said, it's pretty exciing. If capital was unconstrained, which it never is, it'd probably be one that we could also move ahead. But just given focus, we're focusing more on the neuroinflammation, neurodegeneration at this point.
got it. Yeah. No, it's again a very impressive portfolio that spans quite a range of unmet medical CNS needs, and I'd love to have you on an episode maybe next year to update everything, and as these programs proceed and you get closer to commercialization, obviously with the Parkinson's product, but as you progress everything else. But really impressive work, Craig. Again, for everybody who are going to be listening to this particular episode of the show across the various podcast networks, or who will be watching on our YouTube channel. Again, you've been listening to Craig Thompson, Chief Executive Officer, Cerevance, doing really amazing things at the intersection of central nervous system disorders, artificial intelligence, patient monitoring, and all those sorts of cool things. Craig, really interesting story. I wish you guys the best as you move everything forward. Obviously, thank you for sharing the story with us. And as we like to say here on our show, thanks for helping to create a better tomorrow for millions of people out there with CNS disorders. You clearly have an awesome portfolio, and I look forward to seeing how it all progresses in the future.
Great. Love to come back, and great talking to you, Ira.
Yeah, great having you.